Welcome to the Syneos Health Podcast, Early Signals, where we explore the ideas, innovations, and emerging trends shaping the future of oncology. I'm Dr. Wael Harb, Head of R&D and Scientific Strategy in Oncology at Syneos Health. Recorded live from ASCO 2026, these conversations bring together perspective from leaders across biopharma, academia, and technology on the signals they're watching most closely, from AI-enabled drug development and real-world evidence to precision medicine. These conversations offer a snapshot of where oncology innovation is headed next. Let's tune into one of our conversations now. Cell therapy in solid tumors has spent a decade fighting the same two enemies, toxicity and access. TIL therapy proved the biology works. The lifileucel approval made that official. But the next question is harder. Can you make an engineered TIL therapy that's powerful enough to matter and tame enough to deliver outside a handful of academic centers? My guest today has built his career around exactly that question. Dr. Parameswaran Hari is Chief Medical Officer at Obsidian Therapeutics. He came up as an academic hemonc and cell therapy physician at the Medical College of Wisconsin, helped shape TIL clinical development at Iovance during the lifileucel era, and is now leading the clinical development at Obsidian, an OBX-115, an engineered TIL armored with regulatable membrane-bound IL-15 with acetazolamide as the pharmacological switch. Welcome to Early Signals.
Thank you, Dr. Harb.
Take me back to your academic career and why did you decide to switch to the industry?
Yeah, I've been doing cell therapy and bone marrow transplantation, which is the original cell therapy. Over this long career in academic medicine, I was on the effector arm, so to speak, enrolling patients in clinical trials, doing pivotal clinical trials of some of the drugs in myeloma, some of the cell therapies that became approved back in the day. Academic medicine teaches you what patients need. The industry actually teaches you what it takes to build and fulfill that need. So I'm very grateful that I moved to Iovance in the lifileucel era and got a ringside view and was a in-the-ring participant in the clinical development of lifileucel that led to its approval. Then the challenge is always the science. You have to make everything accessible, as you mentioned, everything safer. It's just like the early days of flight. You know, the Wright brothers plane or the Lindbergh plane is not what we fly in now. And that took the effort of hundreds of people to get us to where we are. So it takes time. And now, thankfully, science, the cycling is much, much faster. When I saw the technology, the cyto-drive technology at Obsidian, it excited me. And I realized that this is potentially a technology that can get more patients treated.
Dr. Hari, walk us through your experience at lifileucel and why there's a need to develop another TIL therapy.
TIL therapy is from the NCI surgery branch. Dr. Rosenberg's seminal findings and efforts over the years has come a long way. Lifileucel solved the problem of scalability and the initial problem of access, but it is still dependent on the same paradigm of surgical tumor procurement, lymphodepletion, and the set NCI type of regimen, and then using IL-2 as a cytokine to support. All of those have well-known issues with who can access that therapy. In the days of the NCI, although this was effective pre-ICI therapy for melanoma, many people could not travel there. The access was so limited. Lifileucel, through their scalability, manufacturing success, and availability to patients, and did the monumental effort to get it FDA approved. That actually improved access. Now we need to make it patient-centric so that more and more patients can get it. It can be done in the non-ICU setting. It can be done by not so specialized centers. It can be done on a hematology ward instead of an ICU. It can be done potentially outpatient. Those kind of things are what we need to work on. And I think that's where armoring and regulation have special importance.
Tell us about what you're doing at Obsidian and how this technology is differentiated.
Obsidian's core technology is what we call the cyto-drive platform. And it is a way of post-translational modification of protein or regulation of protein. And what we take is a transgene that produces a protein, but the protein gets destroyed because we are coupling it in the transgene with another protein, typically a enzyme that is purposefully misfolded so that it gets destroyed by the proteasome. So your transgene protein product does not get expressed unless you stabilize the degrader protein. And in the case of OBX115, this degrader protein is carbonic anhydrase, and the ligand for that, as you all know, is acetazolamide or diamox. People take it for preventing altitude sickness. So that drug stabilizes the membrane-bound IL-15, which is coupled to the carbonic anhydrase degrader, and it allows the expression. But don't think of it as a on-off switch. Think of it as a tunable switch. So it's pharmacological regulation, in that if you take more acetazolamide, you get more IL-15. If you take no acetazolamide, the IL-15 stands down. So it's a pretty nice way of controlling cytokine after you infuse the TIL. And it actually does a couple of things. It actually gives you temporal control of when you want the cells to stand up and fight the tumor. It actually allows control for safety. It also allows a one and done cell therapy to have redosing of acetazolamide to actually re-engage the cells over time.
That's very unique because most drug in oncology that we use today, you give the drug and you hope you give the right dose. And we know that there's no two patients alike. So you might not have efficacy because they need a higher dose, or you might have toxicity, so you need a lower dose. But what you develop here is the tunable switch that you can easily, with a pill, to either up or down regulate the cytokine release inside the body.
Exactly. It takes a one and done cell therapy from a one-time infusion. We used to say, pour the cells and pray, to something that's a managed therapy where you're managing the cell effect using a remote control, which is the acetazolamide.
That's a great analogy, using a remote control to control treatment inside the body. That's fascinating. Please give us some understanding where you at in the clinical development. What are the clinical data we have so far?
I would point to the data that was just presented at ASCO this morning by Dr. Betoff from Stanford. So that's our patients treated at the recommended phase 2 dose. These patients were either primary resistant or secondarily resistant to immune checkpoint. Majority of them were actually primary resistant. And they typically have, as you know, very limited options for treatment. These patients were enrolled onto the OBX115 trial and received doses of up to 100 billion TILs based on how much they made. If they made more than 100 billion, they were capped at 100 billion. And then they got acetazolamide in the recommended phase 2 dose, which is 500 milligram pills, 1 week on, 1 week off for the first week 1 and 3, and then they were redosed every 6 weeks. So at that level, we have a objective response rate of 67%, including 2 CRs out of the 15. These responses appear to be durable at this early time point. Potentially, we have to follow those patients for longer. And that is the body of data we have right now.
What about safety? One major issue was lifileucel has been the requirement for hospitalization. You have to give IL-2. It has a lot of toxicity. What have you observed as far as side effects and toxicity?
So our mission has been to develop a patient-centric therapy. There are several aspects to the safety paradigm. Because of this optimized manufacturing process we have, we can actually manufacture TILs from a core needle biopsy. This is our third ASCO presentation in 26. Over the years, we have actually shown that many of our patients have been treated with core needle biopsy, so you can avoid the surgical piece at the beginning. And the advantage is that if you have a liver metastasis or a multiple liver metastasis, you can get at that tumor from an external core needle approach versus having a liver surgeon going. So there's a big difference, especially for some tissue sites. The second part is that we've been able to reduce the lymphodepletion regimen from what's used in the classical TIL regimen of Rosenberg et al., where the Cytoxan dose is what we call the bone marrow transplant levels of dose, 60 milligrams per kilogram times 2. We have been reducing it down to about 45% of that dose. And this regimen is potentially given as an outpatient regimen. And then the most important part is post-infusion, there is no systemic IL-2 or any other cytokine to be used. I think about it as like, if you want to give water to your plants in a garden, you can flood the garden, that's systemic cytokine administration, or you can have a sprinkler that comes on when you need it and feeds the roots of the plant. Ours is like that. The cells carry their own cytokine, and when you take acetazolamide, they turn it on. Right now, all the patient takes is a acetazolamide pill for a week after the infusion, and then a week later, they take a week off, and then take week 3, 1 more week of acetylcholine.
So you really don't need to give systemic IL-2 with your treatment. You're delivering that at the site of the tumor. And more than that, is tunable. You can tune up or tune down to manage efficacy and toxicity.
Absolutely. And that's very important. So we have only one grade 3 CRS out of the 15 patients that we showed today. We have no ICANS. We have no patients that needed ICU level care at all. And the major grade 3 side effects were all hematologic. The non-hematologic toxicities are essentially pretty unremarkable. And the grade 3 hematological toxicities, to an extent, is expected because you're giving a lymphodepleting regimen.
What's next for OBX-115 development?
Current clinical development pathway involves 2 tumor types, melanoma obviously, and the next tumor type is non-small cell lung cancer. There we are several quarters behind melanoma because we started in melanoma and non-small cell was added on as a cohort later. And we are still in phase 1 development there, and data are coming in.
It's fascinating how the idea started decades ago with Steven Rosenberg, thinking about tumor-infiltrating lymphocytes and how we can use it to treat cancer. It takes a lot of research, a lot of trial and error to get to the first commercially approved drug that's helped save lives, but has significant toxicity, but also some complication in access. Now we're taken to the next level, next generation. But that's what it takes. It takes all us as an industry working together to move things forward.
Absolutely. So when I look back at the arc of progress with immunotherapy, a lot of it goes back to the TILs. Dr. Rosenberg's group and the others at the time. To a large extent, our checkpoint success also depends on the presence of tumor-infiltrating lymphocytes already in the tumor. Here we are doing the next step. If those guys are not able to do the job with checkpoint inhibitors, we can take them out, expand the exhausted ones, and give them back. And then we can engineer them now and make them more tuned and, as we put it, remote controllable. So it's no longer one and done. It's a managed therapy across the course of the patient's management. Our job as drug developers is to get our drug approved and accessible and available to the doctors. And I think the field will also figure out the ways to use it, where to place it in the treatment paradigm. And I always go back to my favorite disease or my expert disease, which is multiple myeloma, where, as you see, it was incurable disease. And when I first started my practice, it was about a median survival of 18 months. And now at this ASCO, we have the Majestic 9 data presented where the T-cell engagers, and in relapsed myeloma, we are not even seeing PFS years post-treatment.
Amazing progress. What excites you at this ASCO in immunotherapy and cell therapy?
So this ASCO actually start by saying the plenary session and the amazing success with the drugability of KRAS is the highlight of this. That's the big news of this ASCO. But we should not forget immunotherapy. As a transplanter and a cell therapist, my feeling always is that when you cure these incurable tumors, there is always going to be an immune cell on the other side of the cancer cell. How it gets there could be different. It could be a checkpoint inhibitor that unleashes the cell. It could be a bispecific T-cell engager or a trispecific T-cell engager that makes the meeting happen. It could be a TIL therapy that gives huge numbers of effector cells, allowing them to flood the zone with immune effectors. Or it could be a CAR-T or a TCR-T that is searching for a particular target on a cell. Myeloma is a classic example, curable by any other means. In my mind, always, that was the heme malignancy that could not be cured. That is the heme malignancy that's most close to solid tumors. I think we will see that. In melanoma, arguably, a significant proportion, somewhere in the 40 to 50% range, are already cured with checkpoints. Now, we are going after checkpoint inhibitor resistant population, and actually it lifts the field up for everyone. So I think this ASCO is, despite the precision drug targeting of KRAS, we are also seeing substantial progress made in immunotherapies.
That's really amazing to start talking about cure for solid tumor like melanoma. That's something we would not have dreamt of a few years ago. What do you think the position of cell therapy in general and TIL therapy in the paradigm of all the treatment options today? With immunotherapy, we have a lot of innovation in bispecific and trispecific and ADCs. And some would say the complexity and the cost of cell therapy will be not justifiable. What's your take on that?
These are valid criticisms of cell therapy because it's complicated, it's complex, manufacturing is complex, it's personalized manufacturing at an individual patient level. We are also seeing progress. We actually saw data from another company with in vivo CAR T. In vivo techniques are coming through. In vivo makes it off the shelf. We saw data from allo approaches where you are making universal cells that are agnostic to the HLA disparities between human beings and can overcome the alloreactivity of our immune systems and be used in a target-specific manner, but not a person-specific manner. So the field will solve these problems. I'm very confident of the ability of the field to solve these problems. And ultimately, for any therapy to have the maximum impact, it has to be safe, it has to be accessible to the largest number of patients, irrespective of their baseline status. So you cannot have therapies that can only be for a few patients with a specific HLA type or expressing certain antigens and not certain other antigens. So those therapies have intrinsic problems. So once you get past that and you can access it in a medical and biological way, and then accessible in a financial, and it should be available to every patient in the planet. So we have to actually work towards that. And I think that's what we are always doing. But the highest end of therapy today becomes, in a matter of 1 or 2 decades, accessible to everyone. And again, I go back to air travel. Only the wealthiest people could travel transatlantic. In the 1920s and 30s, the prime ministers and the presidents flew across, right? Now everyone flies across transatlantic. So it actually becomes more and more generalized and population affordable.
What do you see happening in the next 5 years for drug development of cancer?
Again, one of the themes at this ASCO has been the amazing success of global drug development, and it's moving out of America and going globally, and we have a lot of science happening all over the world. And I think that's something that's to be welcomed, and wherever science is progressing, we have to welcome it. And I think that's the trend. In my mind, it's irreversible. There was a statistic that was mentioned many, many years ago that right after the Cold War ended, 98% of new drugs since Second World War had come out of the Western world, the United States and Europe, and then almost 90% from the US. Now that's all changing. The rest of the world is catching up with us, and more people are doing science across the world. So I think it's a good phase for drug development in terms of science part. But then the other part, the regulatory piece, what are the hurdles, what are the bureaucratic issues, what are the costs associated with this, and how many patients can enter these trials at various centers at an affordable manner. For a startup that has limited funding, you need to get early data quickly and keep moving. And I think that's where, as a field, we need to come together with the politicians, with the regulators, and with society to come together to find a faster way, because patients cannot wait and we need to be there for them.
We definitely have to be there for our patient, and it's exciting time that we can deliver meaningful progress for patients with cancer better than ever before. Any final thoughts, Harry, you would like to leave with the audience?
I come to ASCO every year, have done so for a few decades now, a couple of decades plus, I guess. And I go back energized and motivated. I see what everyone else is doing. I see amazing progress. You cannot take away what we saw at the plenary session yesterday and the simultaneous publications that came out, etc. For our patients, I would say that we are all working hard. You, me, we have a collaboration going on in terms of how we are developing OBX115. So let's all get motivated by ASCO and these amazing results.
Thank you so much for insightful and hopeful conversation. To see where the field is going is very exciting for patients. Very exciting time for drug development. Looking forward to continue the conversation in the future.
Thank you so much, Wael.
That's all for today's episode of the Syneos Health podcast, Early Signals. I'm your host, Dr. Wael Harb. If you have other topics you would like to hear on this podcast, please send us a message at podcast@syneoshealth.com. For access to more future-focused, actionable life science insights, please visit the Insights Hub at syneoshealth.com.